There we go. So bear in mind the webinar is being recorded, just in case any interactions afterwards will go down on the record, and we will make the recording available to everybody who has registered for the webinar. Couple of other places you can keep an eye on if this webinar today doesn't cover something that you're interested in learning about from protocols.io. There is a page where we list all of the webinars, and we routinely run introductions to protocols.io, more advanced webinars. And then the second link here is if you would like a one-to-one demo or a demo to a small group of researchers, there's a location where you can sign up for one of those and schedule it at a time that suits yourself. And it would be our customer outreach person who would have that demo with you. I am going to, as I said, try and keep an eye on Q&A through the webinar. So if something's relevant and it pops up and I see it, I may be able to answer it at the time, but if not, I'll have a look there at the end. So we should have time for Q&A at the end. But if you have them actually either in the Q&A or in the chat is fine. That would be great. Please feel free to add them. Okay, so here are the items we will cover today. So first of all, really just framing how and why to think of methods as a modular research output and one that merits credit in and of itself. I will then give you a little bit of an intro to protocols.io. So I am the Director of Strategic Initiatives at protocols.io. So that's the tool that I'm representing today, but I will mention and show you that there are lots of other places where you could publish methods if that is your thing and you want to try to get credit for the method development that you're working on. And then I will show how you can use linking to protocols.io from within research articles to really make the most of this kind of modular publishing, as I like to think of it. And as I mentioned, we will have time for Q&A. I realized I didn't introduce myself more than to say my position on protocols. In a past life, I worked as an editor with open access publisher PLOS. So prior to joining protocols.io at the start of 2020, I was the chief editor at PLOS Biology. So I have spent a lot of time thinking about open access and availability, access to all kinds of research outputs, the articles themselves, but also data and methods, and all of the other elements that you may come across within a research project. So on the general day-to-day of a researcher, we'll be performing whatever kinds of experiments. This Rocky, our raccoon here, is clearly a wet lab scientist with his goggles, but it may be science, research in a different discipline, even social sciences, humanities, in which case lab notebook's not quite accurate, but somewhere you'll be writing down everything that you do from the day-to-day, from designing experiments, running experiments or protocols, gathering data, analyzing data, interpreting, concluding, and the methods that you use. All of those things will be put in some kind of notebook, lab notebook or otherwise, whether paper, as was historically the case, or in more recent times, electronic versions. So how does that look? We have a researcher, again, we're a little bit wet lab biased, researcher here who's generating various different kinds of outputs. So the one that's recognized within academia generally and research generally is the research article as a key currency for researchers. But alongside that, researchers are often generating code, they're certainly generating a lot of data, there are citations that they draw upon, and there will be methods too. So I really want to have you think about research outputs as modular, and why it is that each of those modules is important. So methods in that context, which is what we'll focus on today, really do matter. They're absolutely critical for open and reproducible research. So what I have here, as we can't be together and actually eat nice chocolate chip cookies, we'll just have a photo of chocolate chip cookies. This this photo from a magazine article where someone had made tweaks to the same recipe. So they had just changed something slightly. So if cookies are the data that you're generating in your research on a daily basis, so if cookies are the data, you can only analyze the differences, the phenotypic differences that they have in size, in thickness, perhaps texture, hardness, flavor, these can only be interpreted if you know what the tweaks were to the method. So you have to understand the differences in the method to know why it is that one is larger, one is harder, etc., etc. So the methods are absolutely critical in understanding what happened to get to your data. And we do generate tons and tons of data now. The open research movement has made great strides in ensuring that we have access to a lot more open data now. Many journals are requiring data availability or data accessibility statements. Some are mandating availability of the data when ethically appropriate, and so on. So we have tons and tons of data, but what are we going to do with the data after the fact? So I have a nice quote here from evolutionary biologist Dobzhansky: "Nothing in biology makes sense except in the light of evolution." So my tweak to this is that, oops, I don't know what I've just clicked on. My tweak for that, I don't know why we've gone not quite to full screen. Hopefully that's okay. Is that nothing in research data makes sense except in the light of evolution. So for open science to really work, we do need method sharing. And so when you're sharing your data, you also need to share the comprehensive methods. And my point today really is that the methods themselves are a research output that merits credit in and of itself. So if you're the researcher in the lab with your notebook, you may spend 2 years or so actually honing and optimizing a method before you get around to applying it to an interesting research question. So quite often the development of the method and approach is the most arduous part of the research process. Back to full screen. And so do we do this well? Actually, sadly, no, right now we don't do this very well. So here are a couple of tweets. On the left is a biologist who was looking for a protocol from a 1997 paper, as described in another earlier paper, as described in another earlier paper, and then they hit a paywall and couldn't access the original paper. This is incredibly frustrating for researchers. On the right is the exact same scenario from a physicist who was looking to understand how devices were fabricated, as previously described, and then eventually fabricated with conventional methods. So this makes it really hard for data to be interpreted and also for experiments to be replicated and repeated. Another example which highlights this problem as something that we're not doing well is the Cancer Biology Reproducibility Project. So this was an effort by scientists at the Center for Open Science to attempt to replicate all of the experiments from 50 published high impact cancer research papers. On the left is a write-up about this effort by Ed Yong in The Atlantic, which is a really nice article. And he writes, after talking to the researchers who were involved, that the hardest part by far was figuring out exactly what the original labs actually did. Scientific papers come with method sections that ought to provide recipes for doing same experiments. Often those recipes are incomplete, missing out important steps, details, or ingredients. And in some cases, the recipes aren't described at all. Researchers simply cite an earlier study that used a similar technique, as we just saw in those other tweets. And in fact, if you look up the web page of this initiative on the Center for Open Science site, in their project overview, it actually shows that 0% of the protocols were completely described in the papers that they looked at. And this effort proved to be so time and money expensive that in the end, they shrank the effort to just looking at 18 of the 50 papers because it was not working at all. They were unable to replicate most of the experiments. It turned out, the experimental details were not only not in the papers, but they also spoke to the authors. And the authors didn't have sufficient access to the full details of those experiments either. So repeating and building on those is very, very hard. And this really does serve to highlight that the methods themselves are this critical standalone research output, which deserves credit independently from the analysis in any one scenario. So we'll come back to the modular research outputs. Let's think about methods. How can we capture methods? Are there lots of ways for researchers to do this? What exists right now? So back in 2004, Nature Methods was launched, which was probably one of the first of these, with Veronique Kierma was the editor at that time. And I believe she really had to make a case for this to make people understand that the methods themselves were important and merited publication independently from an analysis per se. So this is from the editorial written in that first issue back in 2004. So contrast, the method sections of specialized journals, although extremely useful for targeted peer groups, do not grant technical information, the wide exposure conducive to interdisciplinary exchanges. Journal addresses the broad audience of researchers working in the life sciences. So there's this understanding finally starting to happen that, you know, availability of the methods is important in a much more interdisciplinary way across all of the life sciences. And then on the right here, I just have logos from other options that exist, other journals. Some are videos, some are open access, some aren't. Different places where you can publish and get credit for your methods. But of course, my focus today will be more on protocols.io, which is who I am representing. So we are an online open access platform for development, optimization and sharing of methods. So our mission is really, really simple. We want to make it easy to share method details before, during and after publication. And with this, we facilitate researchers getting credit, being able to get credit for the method development independently of the research project itself. And for those familiar with FAIR as terminology to describe efforts within open research, findable, accessible, interoperable, and reusable, we tick pretty well all of those boxes as an enterprise and platform/tool. So as mentioned, it's an open access repository. So everything that is published on protocols.io is then available for anyone else to come along and look up and use, reuse, have access to those methods. And that is how then you can get credit for the method on its own. Although started out initially with a focus on wet lab researchers, it has really become much more versatile. And now we receive content across all different research disciplines. It's a collaborative tool. Researchers can come and, you know, work with everybody in their group, or they can work on their own if they want to, but in a way very similar to Google Docs. The researchers have control about how and who they collaborate with and how publicly or privately they perform those collaborations. And the content is archived and mirrored. And I mentioned it's an open access repository, so there's over 12,000 publicly available protocols on the platform today. There's a lot of private content too, and some of this are groups who prefer to keep their efforts private, but some are people who are developing methods, and so this is before they're ready to be shared more publicly. But as you can see from the views and visibility of protocols, those that are available openly are much accessed and viewed. We had well over 1 million visitors in 2021. So publishing content on protocols.io, I am kind of assuming that you know how you would go about getting a protocol onto protocols.io. And so here's one I made earlier, and I'll try and run this little video and hopefully it will work. But my shiny new protocol here is in draft version. And the video will just show you exactly how easy it is to publish that and get a DOI for that protocol. So on the right-hand side, click publish. There is a set 7-step process. So I understand that getting a DOI will make it not editable. We want abstracts for these because the content is indexed after it's published in various different places. We absolutely have to have authors, so at least one yourself, but it doesn't have to be you. We can take funder acknowledgement, so we can put the funders and the grant IDs so that funders can track this. We would like to know if it's a working protocol or if it's one that's in development, if it's a research protocol rather than, for example, a recipe. There are workspaces on protocols.io. You can choose which workspaces this will show up on. So maybe you want it to show up as a coronavirus method. If it's public health, you can add a disclaimer, confirm co-authors and supervisor agree, and understand that this will make it permanently available. And then you will just click publish. You can preview if you want to, and that's it. It's been published, and this protocol now will have a DOI and be publicly available for anyone to see. If you get that prompt, please fill it in and give us some feedback on the process. But I don't know if you remember, at the start it was orange, that's it, and at the end it's blue, and so blue means this is a published protocol. So right now anyone can come along and publish their protocol, and we screen them after they have a DOI. Oh, sorry, my mouse is making it jump around today. So we screen them after they've had the DOI assigned, and we remove any spam or pseudoscience that's come through. We had started it out this way. We weren't getting a lot of spam. It was very rare. Unfortunately, the spammers have now found us, so we are having to take down quite a lot. So coming very soon will be a slight change to that workflow in that there will be a pause in between you clicking publish and us actually getting you the DOI, because we will be screening during that time frame so that we can stop the spam coming through before it's published. Very much similar to the bioRxiv preprint approach, where content is screened before it's posted. This will kind of ensure additional integrity check of content before publication. And that should only add a day or two to the time to receive DOI. It won't really hold things up very much at all. And what can you do then once you've got your protocol DOI? What can you do with it? So what we really want you to do is to link to the protocols as part of the materials and methods in your research articles. So here is an example from a Nature Neuroscience paper. And in the materials and methods, you can see they have brief descriptions of methods, but then they point to the link for the protocols.io protocols for those specific experiments. So this is a brilliant thing, because it may be that the authors of the protocols are not the same author list as the research article itself. So the protocol can have different first author. If it's someone different who's really honed and developed the method, it may be that half of those authors were not involved at all in developing the method. So you can have this completely separate output from the researchers who created and honed and optimized the method, and an independent citation for the method from the research article. So the credit can be separated out, not only kind of for methods versus paper, but for individual methods in the papers as well. So there's a different one for this generation of single nucleus suspensions than there is for fans for enrichment of dopaminergic nuclei. So that's one example from a Nature Neuroscience paper. Here is another example of how this works in a PLOS biology paper. And this PLOS Bio paper, they actually published the protocols as a collection. So in the materials and methods of this paper, it's noted that methods and protocols for all of the experiments are available as a collection in protocols.io, and then the DOI link. And we encourage the DOI link in the materials and methods, but also as a citation in the citation reference list as well, so that the protocols get that credit part in the citation list too. Some journals are not open access, but their references and citation list will be open, where the main content of the paper may not be. So if you click the DOI, it will take you to protocols.io. And this is another place where using a tool like protocols.io gives you a lot more flexibility. You'll come to protocols.io and you will all immediately see if the version that's linked to in the paper is the most recent version of that collection or that protocol. So in this situation, actually, there's a newer version of the collection that's been made available. So what this means is that the authors have been able to come back and either clarify, improve, or slightly tweak or correct a typo or a minor error in the protocols, put up a new version on protocols.io because they can do this whenever they want. And they've not needed to go back to the journal to request a correction. So if you click to view the newer version, it brings you to the latest version. Oops, jumping ahead again. So you'll get the latest version, and that won't be grayed out with any kind of alert over it. So you'll know when you're at the final version. You can also click where it says version 2 here, and you'll see if there are any subsequent versions. It tells you that it's a collection of methods and protocols from the manuscript, but you also see that under metadata for the protocol and protocols.io, there is an individual citation for the protocol collection and the citation for the manuscript. And as I said before, they don't necessarily have the same first authors. So you can allow for different credit recognition for the methods than from the paper itself. So there's that citation again. And just to reiterate that anything you would expect from a high quality journal publisher, we are doing the same with content that's published on protocols.io. So they're all citable. Everything is archived and mirrored in such a way that if anything ever happened to the platform, you would still be able to access the published content. But also if we just had some kind of a blackout with our servers temporarily, you'd still be able to get to mirror of the content on GitHub and other locations. When you use protocols.io, you can group things into workspaces in public groups where content can be organized. You can have discussions with other researchers who join. And it's really got this lovely dynamic functionality. So other users can come along and fork a new copy if they have tweaks to your method for how they can apply it in their lab. I just showed you the versions, but also researchers can put comments on protocols. So we see this happening a lot where a user has a question about how to do something, or perhaps they need a step slightly clarifying, and they can pop a question onto the protocol, and the authors of the protocol will be alerted and can respond to it then and there. So it becomes a bit of a public FAQ. And again, it goes straight to the authors of the protocol itself, rather than if you had a question about a published paper, you may go to the corresponding author who might be, the senior author from one lab of 3 involved with all of the different experiments. So you have a much more direct interaction opportunity with the people who develop the method via the platform protocols.io. And dynamic permanence is how I talk about the fact that you can update methods on protocols.io in a way that you can't come back and do to the published research article without a very formal correction taking place. So, all well and good, you can do this, but do journals and publishers like you to do this? It turns out actually, yes, there are well over 500 journals, several different publishers who strongly encourage their authors to use a tool like protocols.io to share the methods from their research articles. We have a lot of funders also who require or recommend protocols.io in their grant guidelines and policies. And we are starting to have more and more institutions who are partnering with us to provide site licenses to their researchers so that it can be used as a tool more on the day-to-day as researchers develop and optimize methods, rather than just a place for publication at the very end of the process. It's much easier if you've been using it all the way through the research day-to-day process. And then we've, in recent times, built up some additional relationships. So one that we have is with PLOS One at the end of 2020, start of 2021, we launched an initiative where PLOS One added a new research article type, lab protocols. And these are methods papers where the materials and methods are included as protocols on protocols.io. So I'll show you with another short movie here how it is that that works. So this is the first published paper, this lab protocol paper. You can see the protocol link high at the top. If you click to view materials and methods, you can see again the link through to the protocol. And if we jump across, it will take you to protocols.io shortly. And you're brought straight to the methods for that paper. They're bidirectionally linked, so you can see the +1 article. You can see that it's a peer-reviewed method, works for me, indicates other people have been able to get this to work, and the versions and so on. And then that view there with the different steps is so much nicer than a narrative materials and methods that you would see in a normal research article. It's much more appropriate for presenting methods this way. You can view various different things on the tabs there, metrics, views, how many forks, the citation, whether it's been bookmarked by other users and so on. So there's lots of nice ways to track reuse as well of the methods on protocols.io in a way that's, again, slightly harder to do from a paper itself and just citations of research articles. So this just shows you the same thing. And what you didn't see, or perhaps only caught a snippet of, is that there's a PDF of the current version of the protocol that's included as a supporting information file as well on PLOS ONE. But again, you know, the ability for authors to come to protocols.io and update means that the paper itself on PLOS ONE will always point to this version, but the authors can still come back and dynamically update and make new versions and so on. And there was a really lovely thing that happened with this version, because this initial protocol used some proprietary software, and immediately someone came along and made a new fork using some open source software so that it would be accessible to more researchers. So the value of opening up really made a big difference straight away in this instance. This is just to show that, you know, lots of other journals are perfectly happy for protocols.io to be embedded into their journal articles. So on the left, we have a gigabyte paper with an embedded protocols.io protocol collection in the methods. In the middle, it's a current biology paper where the authors are very happy to tell you that they also wrote up the lab protocol for low-cost, high-throughput whole genome sequencing. And then on the right, we also had another partnership last year with Springer Nature, who had published book chapters that were lab protocols. And we have now put all of those lab protocols onto protocols.io so that they are more dynamic for users to interact with and run in the lab if they want to do so. And then in the context of open science and that kind of modular, interoperable publishing, these are just some lovely examples of researchers who are really doing open research already and are now able to get credit for all of these different pieces of output that they're creating through their research efforts. Whoops. So this first one, the paper is here as a preprint, the protocol is on protocols.io, and the code is available on GitHub. So they've really separated everything out. This one, code, protocols, and paper are fully open. So I think is a wonderful examples of how researchers really can do this now and therefore can get credit for every separate piece, and people can reuse the elements separately too. Allen Institute has done a lot around all of this open research as well, as I'm sure you know, and so again they also have protocols.io workspace where they can share all of their content. And then another researcher over here, first serious attempt at open science, and finally the first paper of my PhD. So they've got their preprint and data and code and protocol on protocols.io. And just to highlight another benefit of having the protocols separately from the research article, this is a lovely example. On the top left was a researcher from Chile who was looking for someone with experience doing RNA extraction from primary cortical neuron cultures, so brain cells. after a few tweets, there was a postdoc at UCSF, Elena, who says, from protocols.io, why don't you try this protocol? And when we look at that protocol in a bit more detail, it turns out that it comes from a GigaScience paper, and it was research looking at a 3-spine stickleback parasite, so a fish parasite. So the chances of a researcher working on brain cells finding a paper on a fish parasite and thinking, oh, I bet this is the place that will have the method that's going to be relevant for me, is pretty non-existent. So the fact that the protocol existed independently from the research paper is the reason why it was discoverable for these researchers. So the RNA extraction protocol is clearly applicable to many different model systems, whether they're cell cultures or larger kind of model organism samples that are being used. So there's really a massive increase in the discoverability of the approaches. Obviously, this is brilliant for reproducibility. It can facilitate research connections between people who may be working on completely different things, helps the reuse. And so by making all of these things happen, it really actually enhances the value of the research, as well as the ability of the author to get credit for the method development independently from the research article, because the people reusing this method would need to cite ideally the protocol rather than the paper on stickleback parasites. And so lots and lots of benefits of linking to protocols.io from research papers. And primarily, hopefully today, it's made the point that you can therefore get credit for the methods themselves as an independent research output, which is separate from the paper itself. And that, I think, is my last slide. Yes, so I've kind of lost track of time. 36, that's about right. We've got about 10 minutes. If anybody has any questions, I will stop the share so it's a bit easier for me to track chat and questions, but feel free to pop a question in the chat or in the Q&A, or I'm not sure if you can raise hands. I might be able to unmute people if they want to talk. And also fine if you've no questions, if you want to contact me later or follow up separately, feel free to drop me an email at emma@protocols.io. Great, thanks, Nikki. Never really know if it's a good sign or not if there aren't questions, but it's totally fine if not. We can, I don't like to presume that people are fast typers, so if someone's still typing, I don't want to cut you off, but if not, we will end there in a mo, and well, I'll stop the recording